In my last video, we talked a bit about siderophores, exotoxins, and direct damage that pathogens exert on human hosts in order to cause disease. In this video, we want to look at the role of endotoxins in causing disease, and we'll also look at portals of exit. Your objectives for this video are to explain what endotoxins are and what microorganisms produce them. You'll also want to be able to talk about or describe their functions and the diseases that they cause. Finally, you should be able to describe what portals of exits are and give some examples.
Your second type of toxin is called an endotoxin. You'll recall I told you that this is part of the actual bacterial cell's structure. So remember way back in chapter 4 when we talked a bit about the gram-negative cells and how they have this outer membrane, and embedded within that outer membrane is a toxin called lipid A. This is what we're referring to. So lipid A is an endotoxin. It's part of the structure of the bacterial cell. As long as the bacterial cell is living and the outer membrane is intact, the lipid A is harmless to a host. But once the bacterial cell dies and starts to break apart, that lipid A gets released and it can cause some pretty significant symptoms. So your endotoxins, their source is gram-negative cells. They are part of the outer membrane. Okay, it is a lipid, that's the lipid A. Now, what do your endotoxins cause? Any ideas? Well, they can cause a variety of different effects. Many of them are very similar to what we see with the superantigens. So things like fever and shock, for example. Your endotoxins cannot be denatured, so we can't make toxoids, which means we can't make antitoxins to the lipid A. So there is no antitoxin for these endotoxins. And as far as an LD50 is concerned, you need a lot of endotoxin to actually cause disease and death. So that's a difference between the endotoxins versus the exotoxins. So as I mentioned before, endotoxin tends to be released when gram-negative cells die inside of a host, and they lyse as a result. This causes disruption of the outer membrane. We also know that endotoxins can be released when cells, gram-negative cells, are multiplying in a host, and certain antibiotics can also cause these types of cells to lyse and release endotoxin. Now, what happens when these gram-negative cells are engulfed or phagocytized by cells of the host, like macrophages? Well, there's a variety of things that can happen. We can get fever, chills, weakness. In severe cases, there can be shock. And in the case of pregnant women, this endotoxin can cause miscarriage. One of the effects that we see is that endotoxin causes the formation of small blood clots in the capillaries, and those blood clots can actually block the capillaries, cutting off blood flow to tissues and leading to tissue death. This is called disseminated intravascular coagulation, and it can be very harmful to a host. Another effect that we sometimes see is called endotoxic shock. Here you have a dangerous drop in blood pressure. And it happens because when these gram-negative cells are phagocytized, macrophages start to release chemicals. And one of these chemicals is called tumor necrosis factor. So if you look at this figure here, you can see a gram-negative cell that's being engulfed or phagocytized by a macrophage. That's what's in figure 1. In part 2, the bacterial cell, is actually being digested. And as part of that process, the outer membrane of the bacterial cell is being disrupted. This releases lipid A. Lipid A triggers the macrophage to start producing cytokines. Remember what those are? Cytokines, remember, are chemicals that are going to disrupt, or not disrupt, I'm sorry. Cytokines are chemicals that kind of modulate and control immune response and communication among cells of the immune system. So here, the endotoxin causes this macrophage to start producing two types of cytokines, IL-1 and TNF-alpha. So IL-1 stands for interleukin-1, and TNF-alpha, that stands for tumor necrosis factor alpha, and they can have a variety of effects in the human body. If perchance these are released due to the presence of bacteria in the cerebrospinal fluid, they can cause weakening of the blood-brain barrier, which allows bacteria to actually penetrate, bacteria from the blood to actually penetrate into the brain and cause damage. You have a few bacteria that are responsible for this. So S. typhi can do this. It causes typhoid. And Neisseria meningitidis can cause meningococcal disease in this way. Now, the release of IL-1 and TNF-alpha can also cause fever. So when these two cytokines make their way into the blood, they travel via the blood vessels up to the brain. And you might remember from AMP2 that the hypothalamus of the brain is kind of like the thermostat of the body. It sets your body temperature. So what happens is that these IL-1 and TNF-alpha trigger the hypothalamus to produce prostaglandins, another type of chemical messenger. The prostaglandins reset the body's thermostat, that's the hypothalamus, to a higher temperature, which causes fever. And remember, fever is one of the symptoms of endotoxin release inside of a host. Now, certain drugs that are used as therapeutics, for example, the humulin that we talked about, remember that was the insulin that's produced by bacteria. We talked about it back in chapter 1. So when certain drugs are produced using gram-negative bacteria, those drugs have to be purified away from the cellular components of the bacteria. If that drug were to be contaminated by endotoxin, and then it was injected into someone who's already sick, it could cause severe disease for that person who is being treated by the drug. For this reason, a special kind of assay has been developed to detect the presence of endotoxin in recombinant drugs, or drugs that we synthesize with bacteria. This is called the LAL assay. LAL, or LAL, stands for Limulus amoebocyte lysate assay. So Limulus, this is the genus name for horseshoe crab, and amoebocytes are white blood cells that we find inside of horseshoe crabs. So what happens is that labs will draw the blood of horseshoe crabs, and they'll spin it down to get white blood cells from it. Now, when amoebocytes, these white blood cells, come in contact with endotoxin, it causes the amoebocytes to actually lyse or break open, and that produces a clot. So what this means is we can take a little bit of the drug that was produced in a gram-negative bacterium and then purified. We can expose it to these amoebocytes and look for their lysis and the production of a clot. If we happen to see a clot, then we know that the drug is contaminated by endotoxin and needs to be further purified before it can be used as a drug or a therapeutic. And if there's no lysis, then we know that the drug is safe for use as a therapeutic in humans. So this is kind of a gift from our friends the horseshoe crab that helps us detect endotoxin. So we began this series of lectures talking about portals of entry. That is the ways that microbes actually get into the body. Remember we talked, for example, about how they can come in contact with the mucous membranes of the respiratory tract or the GI tract. We talked about how they can penetrate the skin via follicles or pores, and then they cause different diseases. We talked about the parenteral route that delivers pathogens below the surface of the skin. So just like microbes enter the body through preferred routes, they can also leave the body through preferred routes. And usually they're going to leave via some sort of secretion or discharge or shed tissue coming from these exits. The portal of entry is related to the infected body part where the pathogens are growing and reproducing, and oftentimes the portal of exit is the same as the portal of the entry. So on this slide, I've actually given you some examples of ways that microorganisms can leave the human body. For example, when tissues of the respiratory tract are infected, coughing or sneezing can lead to the projection of microbes from those infected tissues out into the environment where, those microbes on, say, saliva droplets can then be inhaled or they can be touched or contacted by a susceptible host. From the intestinal tract, microorganisms often leave via the feces or saliva. So, for example, polio is well known to be transmitted via the gastrointestinal tract well after the person who was infected actually stopped showing symptoms. Via the genitourinary tract, microbes can leave the body via urine or vaginal secretions, or if we're talking about a male, via urethral secretions. From the skin, wounds and drainage from wounds of the skin can lead to the exit of microbes from the body. And then from the blood, usually that requires some sort of parenteral or puncture. So biting arthropods or needles, syringes, etc., they can lead to the exit of infectious microbes from the blood. So on the next slide, you see kind of a summary of the things that we've kind of talked about. Hopefully this will be useful to you in your future careers. If you have any questions, you can ask me in class. I'll see you guys next time.
What I hope you got from this video is that endotoxins are toxins that are actually part of the cell. They're part of the outer membrane in your gram-negative cells, and specifically we're talking about lipid A. Remember that your endotoxins have a variety of different effects on human hosts. One thing that they do is they cause fever. Another thing that they do is they can cause a reduction in white blood cell count. They cause the body to release a number of signaling molecules, and all of this kind of accumulates in a drop in blood pressure, which can be a pretty significant event. It can be life-threatening. We also talked a little bit about the different portals of exit. So these are ways that microorganisms leave the human body. They can be the same as the portal of entry. An example would be, say, with influenza, the portal of entry would be through the respiratory mucosa when we inhale viral particles on air or on droplet nuclei. They attach to the tissues in our nasal and in our nasal cavity and in our oral pharynx, and they begin the infection. Well, that's also how they leave. When our nose starts to run, that mucus carries virus particles with it. When we sneeze, cough, or talk and we're sick, that virus that's occupying the respiratory mucosa becomes mixed with saliva and mucus, and we eject that when we talk, cough, and laugh, whatever. So your portals of exit can be the same as the portals of entry, and again, it's just the way that the microbe leaves the body. So that's it for this video. And yeah, if you have any questions, please don't hesitate to reach out with questions. This will be on your third exam, so you'll want to be sure that you're familiar with this video as well as the last video for this chapter. In the next video, we'll be talking more about microorganisms and their interactions with human hosts. I'll see you there.